morning, students. I will tell you about plasma membrane. Cells remain surrounded by a membrane which is known as plasma membrane. The plasma membrane is an ultra-thin, elastic, living, selective transport barrier that encloses the content of the cell. Electron microscopic studies reveal that the plasma membrane is a limiting membrane which serves as a link between the interior and exterior of the cell. It has a definite physical and chemical characteristics with the inherent property of regulating the flow of selective materials through it. History. Plasma membrane was discovered by Overton in the year 1899. In 1855, it was called cell membrane by C. Nageli and C. Kramer. J.Q. Plough in the year 1931 called it plasma membrane. Plasma membrane is present in all types of cells. In plants and bacteria, it is present in between the cell wall and the cytoplasm, while it forms the cell surface for animal cells. Thickness of plasma membrane varies. Average thickness is 75 angstrom. Membrane of intestinal epithelial cell is 85 angstrom, while that of RBC is 215 angstrom. To know the biochemical characteristics of plasma membrane, we need to isolate it. Generally, plasma membrane is isolated from RBC. For this, RBC is treated with hypotonic solution, a solution having lower concentration of solute. When a cell is kept in such a solution, then it swells up and finally bursts. Contents are released, and what left behind is the plasma membrane. In case of RBC, it is regarded as the ghost of RBC. Now we have plasma membrane. The biochemical examination of the ghost membrane or the plasma membrane shows that it is composed of lipid, protein and carbohydrate. I will tell you about all the three components of plasma membrane. I'll start with lipid. Lipids serve primarily as a structural framework for the membrane and as a barrier preventing the indiscriminate movement of water-soluble materials. It constitutes about 40% of the total weight of plasma membrane. For biochemical study, it can be extracted by using organic solvents like chloroform, benzene, ether, etc. The lipid components are of three types: phospholipid, glycolipid and cholesterol. Phospholipid. They are amphipathic molecules, that is, molecules having both the groups positive as well as negative. Phospholipids are elongated molecule. In mammalian cells, they are of about 100 types, asymmetrically distributed. Some are located on the outer surface in contact with the extracellular fluid, while others on the inner surface in contact with the cytoplasm. Structure of phospholipid. You can see here that the phospholipid has got a head and tail part. Head or the hydrophilic part or the polar end of phospholipid. Head is formed of choline phosphate. Tail or the hydrophobic part or lipophilic part or the non-polar end. Tail is formed of two molecules of fatty acids whose carboxyl end connect with glycerol backbone, which in turn with phosphate backbone of head end called hydrophobic tail. The diagram shows distribution of phospholipid. Phospholipids are asymmetrically distributed. This I have already told you. Here in this diagram you can see that sphingomyelin and phosphatidylcholine are present on the outer surface, while phosphatidylethanolamine, phosphatidyl'inositol and phosphatidylserine are located on the inner surface in contact with the cytoplasm. Types of phospholipid. There are two types of phospholipid: neutral phospholipid and acidic phospholipid. Neutral phospholipids form major portion of the membrane phospholipid. They are uncharged molecules. Constitutes 82 to 95% of the total phospholipid. They remain in the bilayer with cholesterol. Acidic phospholipid. They are negatively charged. Constitutes 5 to 20% of the total, and they remain associated with proteins by way of lipid-protein interaction. Functions of phospholipid. Phospholipids form bilayer that provides barrier to diffusion of polar solutes. Example: phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine. They are present in most membranes. Others like lecithin, cerebroside. They also provide barrier to diffusion of polar solutes. They are present in the membranes of nervous tissue. Phospholipids activate cytochrome. Example: cardiolipin. It is present in the mitochondrial inner membrane. Then phospholipids are the source of inositol triphosphate. Example is phosphatidyl'inositol. It is present in most membranes. Now, glycolipid and cholesterol, the other types of lipids present in membrane. Glycolipids are present only in the outer lipid layer. They are the major lipids in the thylakoid membrane of chloroplast. Cholesterol, they are amphipathic molecule. They are present in association with phospholipid tail. They provides rigidity to tail and mechanical stability to the lipid bilayer. This is all about lipids.